Okay, so good morning, everyone. So today we are going to talk about transport in plants. So I'm going to start with structure of xylem and phloem. So xylem is actually transporting water and inorganic ions. And then for phloem, they are carrying organic substance. So just to recap, this is a root stem of a plant, and here is the epidermis, and here is the cortex of the plant, and middle we have the pit, and there's a cambium here. So you can see that cambium is separating xylem and phloem, and for xylem, there's actually made out of 4 cells, which is xylem vessels, parenchyma cells, sclerenchyma cells, and also trachea.
Xylem vessel is made up of
Xylem tissue. Xylem
yeah.
tissue.
So if you cell is xylem vessel element, I think.
Yeah. And then these cells have no cell content inside, so that is in order to reduce the resistance while transporting waters. And these cells is also known as dead cells. This is because that there's empty space in the middle, which known as our lumen. And then this xylem element, you can see that it's joined end to end and form a continuous tube. There's actually more here. So what do xylem vessel made from is actually began as a normal living cells with cellular cell wall, and then which get elongated. As you can see here, the thickening of wall with lining. This one, the lining is actually made of a cytoplasm. The cytoplasm breaks down, and then the wall becomes thicker with a substance called lignin. And this lignin can prevent the xylem vessel from collapsing. And then this tube is hollow so that there's less resistance to flow of water, and it has a large lumen, large diameter, in order to transport a large volume of water. There's also lack of end wall, so that there's less resistance to water flow, and there's pits over here. As you can see, this dot here is actually pits, and these pits allow lateral movement of xylem vessels. Okay, so going back to phloem, phloem is actually made out of two, consists of 2 cells. This part is sieve tube. Well, this part is actually companion cell. So what is the difference between sieve tube and xylem vessels? Is that there's actually cytoplasm in here, but then there's no cytoplasm in xylem vessels. But then there's no nucleus in sieve tube as well. No nucleus, ribosome, and also Golgi apparatus. But then there's actually mitochondria and also ribosome in companion cell. Why is it? It's because that this companion cell have to carry out active transport, so that it requires energy from mitochondria to produce ATP. Okay, so what is the how does sieve tube adapt to its function? It's actually that sieve tube has sieve plate with pores. This is the sieve plate, and you can see there's pores. This sieve plate allows easy flow of material from one sieve tube to another. And as teacher mentioned before, there's actually another function that it holds the wall of the sieve tube together to avoid that it expands and collapse. And then have sieve plate, it can prevent a sieve tube element from bulging under pressure, like prevent from bursting. And there's numerous plasmodesmata with companion cells, a lot loading of sucrose. Sucrose is actually transported out of the cell into companion cell, and sucrose transport into the sieve tube through this plasmodesmata. And yeah, have little cytoplasm. So I'm going to continue with the past year questions. So for the first question, figure 4.1 shows transverse section of a root and a stem. Shade in an area in the transverse section of the root where there are cells specialized for the transport of water. So we have to shade, this is the root and this is the stem. We have to shade the part for where the water is transported. So just a recap, this part is actually endodermis. This part is phloem, and this part is actually xylem. And I mentioned just now, xylem is to transport water and inorganic ions, so that the answer for part 1 will be this part. And next, shade in an area in the transverse section of the stem where there are cells specialized for the transport of sucrose. Sucrose is an organic substance, so it will be transporting in phloem. So this part is actually phloem, and the inner side is xylem. This middle line is known as cambium. So you can shade all the phloem, or you just shade one is enough. And for question B, suggest why the vascular bundle in the stem are situated towards the outside. This is actually to provide strength and provide support to the stem so that it won't break, or because there's wind blows, right? The tree will be moving left and right, and this vascular bundle will provide support to avoid it from breaking. And the answer scheme reject lignin unqualified and reject prevent collapsing. Okay, next question. For figure 3.1 shows an electron micrograph of some xylem vessels in tubercle leaf fragment in the cigarette. So this is a picture of xylem vessels. Xylem vessels. And the questions ask, describe and explain two features of the xylem vessels visible in figure 3.1 that are adaptation for the transport of water in plants. So for this question, I mentioned just now, this is adaptation for xylem vessels. But then for this question, we cannot use all of the above because we only can explain and describe what we see in the diagram. So you have to explain two features of the xylem vessels. So I'm going to copy this first. Okay, so for this question, we can explain that this xylem vessel is hollow. And for explanation, we can explain that it's little resistant to flow of water. And we can also explain that there's large lumen because it's what we see here have a large diameter and is able to transport a large volume of water. But then thickening wall with lignin prevent from collapsing. This is something that we cannot see because we don't know that this has lignin or not. So we cannot use these points for these questions. And lack of end walls to form a continuous tube. This is also a option that we cannot choose because we can't see that is there any end walls at behind or what is not visible from this diagram. And have pits, you also can't see that there's any pits in this diagram, so you cannot use this point as well. So here is a mark scheme. The mark scheme states that it's tube-like, tubular, hollow, empty, or no obstruction, and no cells contained, so that the explanation is so little resistant and easy for the movement of water. And this will reject max flow and continuous column, as I mentioned just now. And another point for another feature is wide, large diameter, or large lumen. For the explanation is that so large volume or large amount of water can be transported. Oh, sorry, just now the thickening of wall, this lignin thickening of wall is also accepted. Sorry. And next question, for figure 5.1 shows a vascular bundle from the stem of Peperomia, a plant from Brazil, and the vascular bundle in the stem of P. destilti are unusual because they are surrounded by an endodermis with a Casparian script. So for this question, use label lines and letter P, Q, and R to identify the following in the vascular bundle. So for P, it's actually an endodermal cells with a Casparian stripe. All this is actually endodermal cell. As you can see that this line is the Casparian strip. So you can just label it P. And a cell strengthened with lignin, just for information that Phloem does not have lignin, but only xylem vessel have it. So this 4 is actually xylem vessels, and this is answer for Q. And R, a tissue that transport assimilate. This tissue means that phloem tissue. And since it's mentioned that it's phloem tissue, so that we have to circle the whole part to show that it's a tissue. And how I know this part is phloem tissue is because that you can see there's one big cells and one small cells. This big one is sieve tube, while this small one is actually companion cell. We can recognize phloem tissue by this way, finding a big cells and the small cells which attach together. So for this one, I will label it as R. Okay, so the answer scheme is given here. P, you just have to label one of the endodermal cells, and for Q, you just label the cell wall of one of the 4 xylem vessels. And then for R, the answer scheme also accepted one cells, but then it's better that you circle whole cells because it mentioned a tissue. And for C, state and explain 2 ways in which the structure of a phloem sieve tube is adapted for the transport of assimilates. Do you want to try for this question? I already mentioned just now.
In Teng, I was thinking that just now that question asked the student to label the tissue, right?
Yeah.
So if the student label the cell, I will not give any mark.
So should be rejected.
Yeah, even though the mark schemes accept one cell, for me, if I mark this question, I will not give mark because cell and tissue are totally different things. Yeah, I'll be more strict in marking. Okay. Yeah, okay, let's look at next question. Thank you.
So do you want to try for this question? Structure of phloem sieve tube is adapted to transport of assimilates.
Yes, you can call their name, it'll be faster. And they cannot sleep and they cannot run away. So call their name.
Okay. Peish, do you want to try?
They have the sieve plate to prevent the sieve tube from collapsing.
Yeah, correct.
A plus 2 marks.
So you can see from mark schemes, there's sieve plates which can prevent the sieve tube from bursting. And there's actually sieve pore, which also allows the easy flow from sieve tube to sieve tube element, which at the middle of both sieve tube. And there's actually self-surface membrane and plasma membrane, which prevent the loss of sucrose at the phloem tissue. You can see from here, this cell surface membrane, it prevents sucrose from transporting to cambium or to the xylem at neighboring. And there's also little content because sieve tube doesn't have nucleus, ribosome, or Golgi apparatus, and this have reduced the resistance for the sucrose to flow. And plasmodesmata, which allows the companion cell, the sucrose to flow from companion cell to the sieve tube. And there's also thin wall, which is easy for entry of water. So that's all for here. I will pass to Januli first.
Thank you, Teng, for the clear explanation.
So today I'm going to discuss some questions about transpiration, xerophytes, and a bit about the Symplus and Apoplast pathway. So here there's a mind map which discusses some of the components of this chapter. Yeah, so as you can see, for the absorption of water, so we know that, just a recap, soil water can enter the root hair cells through the partially permeable membrane down a water potential gradient. So then this water must move across the root to the xylem vessels in the center of the root. So there are two pathways, which is the Apoplast pathway and the Symplus pathway, which I will be discussing further later in the questions. And then we also have the mass flow hypothesis, which is for the transport of sucrose and other soluble organic materials. So yeah. And also for the uptake of transport and mineral ions, please ignore the second box, which is the NPKS, because that's not in the syllabus. So yeah. The next question. Thank you. Okay. So the first question, figure 1.1 is a diagram for from a photomicrograph of a transverse section through a part of a leaf. The arrows in figure 1.1 shows the movement of water through the cells of the leaf after it has left the xylem vessels. Can you scroll down a little bit? Thank you. Okay. So the question says that water from the xylem can enter cell A and then move to cell B and cell C without crossing the cell walls. So these structures are not visible in the diagram that they have given. And the question asks us to name the cell structures through which water can pass from cell A to cell B without crossing the cell walls. So it's the plasmodesmata. It's quite straightforward. So yeah. Next question. Okay. So the next question asks to explain what causes water to move from cell B to cell C. It's also quite straightforward. So we can say that the water from cell B will move to cell C down a water potential gradient. And we can also, the mark scheme also accepts from a lesser negative potential, which is cell B, to a more negative potential, which is cell C. So the next question says, name the pathway taken by water between cell A and cell C. As in the previous question, we know that water can move from one cell to another cell through the plasmodesmata. So this pathway is known as the Symplus pathway because it doesn't, the water doesn't move by the means of the cell wall. So next question. Okay, so The next question says that most of the water which arrives at the leaf passes to the external atmosphere. With reference to figure 1.1, describe and explain the sequence of events occurring between point P and point Q. So point P is basically the cell walls of the spongy mesophyll cells, and point Q is the atmosphere. So the question is basically asking us to describe the sequence of events which takes place in transpiration. So for the first point, we can say that water evaporates from the large surfaces of the cell walls of the spongy mesophyll cells. And then this water vapor will diffuse across the intercellular air spaces and substomatal air spaces, and then it will diffuse out of the leaf to the atmosphere via the stomata. So as you can see in the marking scheme, they say reject palisade mesophyll cells, and this is because the evaporation of water is at spongy mesophyll cells and not the palisade mesophyll cells. And the question and the mark scheme also says reject diffusion of water vapor. If you write out of guard cells, they will also reject it because water vapor diffuses out of the leaf via the stomata. So yeah. And also the mark scheme accepts the symbol for water potential, which is psi, but I think it's better to write out water potential instead of writing the symbol. So yeah, next question, please. Okay. So the next question is about xerophytes. So marram grass is an important plant of sand dunes. Leaves of marram grass are well adapted to reduce water loss by transpiration. Figure 2.1 is a photomicrograph of a section through the leaf of marram grass. Some examples of adaptations to reduce water loss by transpiration includes thick cuticle, no stomata on the outer surface, and stomata in pits on the inner surface. So the question asks us to state one other adaptation which is visible in the diagram that they have shown, which reduces water loss by transpiration. So we can say that there's curled or rolled leaf and also the presence of trichomes, which are the hair-like structures in the lower epidermis of the leaf. So in general, some other adaptations of other xerophytes includes reduced numbers of stomata, or the stomata can only be found in the upper epidermis. And we can also say that the stomata can close during the day and open during the night to reduce the water loss by transpiration during the day and also to prevent the plant from wilting. And also sunken stomata and the presence of stomatal crypts. Next question. Okay. So the next question asks to explain how the adaptations reduces water loss. So the adaptations which we have given in the first question are the curled leaf or the curled leaf and the trichomes, which are the hair-like structures. So we can say that the curled leaf reduces the surface area of the leaf. And the curled leaf and trichomes also creates a humid airspace in the middle. And this can lower the water potential gradient between the airspace and the internal surface of the leaf. So it reduces the water loss by transpiration as well. And as the question has stated, there can also be thick cuticle. So we can say that the thick cuticle actually increases the diffusion distance. So the water molecules will have to travel a much larger distance, and therefore the rate of diffusion of these water molecules will decrease. So yeah. Next question. And the last part of that question says to state the term that is used to describe a plant type that has adaptations to reduce water loss by transpiration. So it's xerophytes. It's quite straightforward. So, okay. So this question is about the Symplast and Apoplast pathway. And the question says that water absorbed by plant roots travel by different pathways from the root hairs to the xylem. Figure 6.1 shows these pathways in the root of Ranunculus acris. Okay, so the solid line represents pathway B, which, as you can see in the diagram, it only passes from the spaces in the cellulose cell walls, while pathway C, which is the dotted line, it passes from the cytoplasm and then from in between cells through the plasmodesmata. And as you can see in the diagram, the endodermal cells have there is a thick Casparian strip, which is made up of a waxy and waterproof material called suberin. So from the endodermal cells, the water must travel to the xylem vessels via the plasmodesmata, because it cannot travel through the cell wall due to the Casparian strip. Can you scroll down a bit? Thank you. Okay. So name the question asked to name cell A and pathway B. So cell A is actually the endodermal cells, and pathway B, which is the solid line, is the apoplast pathway, because the water moves from the root hair cells to the endodermis via the cellulose cell wall. So it's the apoplast pathway. Just a quick recap. Basically, apoplast pathway is a diffusion of water through a series of air spaces in the cellulose cell wall. So they do not cross the partially permeable cell membrane. While the Symplast pathway is a movement of water via it can move from the cytoplasm and enter the vacuole through the tonoplast, and also move from one cell to another cell via the plasmodesmata. Okay. Okay, so the next question is about transpiration. So it says that transpiration occurring at the leaves is mainly responsible for the movement of water across the root of R. acris. So explain how transpiration is responsible for the movement of water across the root as shown in figure 6.1. So we know that basically, so water from the xylem vessels, it can pass out of the xylem vessels to the mesophyll cells, and this results in a lower water potential at the xylem vessels in the leaf. So the xylem vessels in the roots will have a higher water potential, so the water is being pulled up the xylem vessels down a water potential gradient. And since the question says that is due to transpiration, so we know that transpiration is basically the diffusion of the water vapor out of the leaf via the stomata. So we can say that this creates the transpiration pool, as you can see in the mark scheme, and it also creates the tension or the suction force, which is used to pull water up the xylem vessels. And we can say that water can move up the xylem vessels by cohesion-tension. So we know that cohesion is actually the attraction between water molecules due to the hydrogen bonds which can be formed between them. And we can also say it's the addition between the water molecules and the cellulose in the walls of the xylem vessels. So yeah, the question is actually 5 marks, but the marking scheme has given 10 different points. So you can just write 5 points from what they have given in the mark scheme. So yeah, next question. Okay. So the next question asks to explain why the movement of water in pathway C is slower than in pathway B. So pathway C is the simplest pathway because the water moves from one cell to another cell via the plasmodesmata, and pathway B is actually the apoplast pathway because water can it basically moves through the air spaces in the cellular cell walls. So we can say that pathway B is much more faster because in pathway C, there is much more resistance because it has to travel from the cytoplasm and through the plasmodesmata from one cell to another cell. So it's much more faster compared to pathway B, which is the apoplast pathway. Because diffusion through air spaces is much faster than diffusion across the plasmodesmata. So yeah, that's all for my part. Now I'll pass it back to Yin.
So can I add a little bit more on this simplest pathway that's slower based on the mark scheme? So the water is passing through cell surface membrane. If it first enters the cell, it has to go through the cell surface membrane, so slower. Then inside the cell, it might also pass through the tonoplast. So when it passes through the tonoplast, another 2 layers of membrane it need to pass through, so that gives a high resistance to flow. And then, of course, if you talk about movement of water in cytoplasm versus movement of water in the cell wall, of course, movement of the water in the cytoplasm will be slower because cytoplasm contains a lot of other biological molecules, so that the liquid should be thicker, so it gives a greater resistance to water flow, versus if the water flows in the cellulose wall, where basically it's just the water that coming in from the soil. So it could just be pure water with some mineral ions, versus in cytoplasm, there'll be water, mineral ions, and also organic molecules like carbohydrates, proteins, and so on. So the resistance to water flow in the cytoplasm, even though it's moving from cell to cell through plasmodesmata, is also through the cytoplasm, right? The cytoplasm itself. Plasmodesmata is a cytoplasmic strand. So because of that, the movement by simplest pathway will have got a higher resistance than the movement in apoplast pathway. Okay, thank you.
Thank you, teacher. So now I'll pass it to Ying Teng. Okay.
Okay, so I'm going to continue with the part for translocation. For recap, translocation is divided into 3. I'm sorry. Translocation is divided into 3 parts. The first part is the loading. Second part is translocation. And the third part is unloading. So what is translocation? It's a transport of soluble organic and inorganic substance from the source to the sink. And the example for the source is leaf, onion, carrot roots. And for the sink is actually roots, fruits, flower, or other region of growth. And what is the assimilation? Is actually sucrose and amino acid. There's also other plants growth hormones which will be transported. And there's and for the first step, before that, I will go through another question first. Name G and H. This figure 2.1 shows part of a summer squash, Cucurbita pepo. Figure 2.2 is a high-power drawing of an area of phloem from a transverse section of the stem of sea pepo. So this is a transverse section of sea pepo, and name G and H. Do you want to try for G and H? You can just type in the chat. It's okay. For G, since the cells is big, it's actually sieve tube. And for H, there's a small cells beside a sieve tube. So this is a companion cell. And you can see this part is actually plasmodesmata. And describe 3 ways in which the structure of a xylem vessel different from the structure of cell G. So we look at the mark scene. Xylem vessels actually have thicker wall. Sorry, it's lagging. Xylem vessels have thicker walls and thickening in walls. This is a slide from teacher's PowerPoint. As you can see, there's 5 types of lightning. First one is annular, spiral, scalariform, and reticulate and pitted. This reticulate is actually net shape. And so in phloem vessel, in phloem tissue, they does not have lightning, so there's no thickening in wall. And xylem vessel have a wider lumen compared to a phloem tissue. And xylem vessels does not have cytoplasm, and their reject date is because that cell is not the structure of a xylem vessels. And xylem vessel has also have pits and no cross wall, no sieve plate, no sieve pore, unlike phloem tissues. Sorry, I'll change Wi-Fi for. Okay, for B, this question I will skip first because I will further explain at behind. And for C, most of the sucrose transported in phloem enters the fruits, suggests why summer squash fruits are not sweet. So we look at the mark schemes. Sucrose are used in respiration. They will be converted into glucose and fructose, which are needed in respiration. And it will also be stored as starch and used to make cellulose. They also accept used to make cell walls, as the sucrose will change into cellulose, and cellulose is one of the components of cell walls. Store as or convert to organic acid in workers and convert into name other substance like lipid or proteins. Okay, so I'm going to explain the loading for companion cells for translocation. So first, these companion cells will use the ATP Okay, imagine this dot is a proton pump, and there's actually a lot of H plus inside compared to outside. Thus, there's lower H plus compared to outside. So when you transfer from a lower concentration gradient to a higher concentration gradient, this is active transfer and you require ATP. So this ATP will break down into ADP plus phosphate, and this phosphate will bind to the proton pump to allow the H plus ions to actively transport out of the companion cells. Then these H plus ions will diffuse back in to these cells by diffusion as from a higher gradient to a lower concentration gradient. So
No. From a higher concentration to a lower concentration down the concentration gradient. Higher concentration is like the top of the hill. Lower concentration is at the bottom of the hill. So the gradient is going down the gradient. Okay, not from a gradient you go to another gradient. So you're not from a hill go to another hill.
Okay.
Okay.
From you move down the concentration gradient. So this sucrose, the concentration gradient of this sucrose of outside is higher than inside. So it is outside is lower than inside. Okay. Yeah.
What is lower? What is lower? Say the whole thing.
The sucrose outside of the cell is lower than the sucrose inside the cell.
No, cannot lower. Is the number lower or concentration lower?
Concentration lower.
Okay, say the whole thing again.
The concentration of sucrose outside of the cell is lower than the concentration of sucrose.
Correct. Inside the cell. Very
outside
good.
is lower than inside of the cell. Yeah. So it requires you have to co-transport together with H plus ions through a proton sucrose carrier protein. So we co-transport together with H plus. This you have to note that is sucrose co-transport together with H plus, but not H plus co-transport with sucrose. So when they enter the cells, the water potential of the cells will drops, and this causes a H2O from neighboring cells to enter the companion cells through osmosis down the concentration, down the water potential gradient, and this also include then this will cause a hydrostatic pressure inside these companion cells is higher, is built up and push this sucrose through this plasmodesmata into the sieve tube. So now the sucrose already entered the cells at the sieve tube. So now we will go to translocation. Translocation of sucrose will make the water potential of sieve tube to drops, and the water will enter by osmosis from the surrounding cells, like this H2O will enter from here, and the xylem vessel, the water will also enter into this sieve tube. So this hydrostatic pressure will increase, and imagine this part is a source. So the hydrostatic pressure increase will make it higher than the hydrostatic pressure of the sink. And this will make the sucrose to be pushed down the pressure gradient and move into the sink. And this is also known as mass flow. And after that is unloading. So you imagine now we are at the sink. Here is the sink. So since unloading of sucrose requires energy, so the sucrose is here. Here have higher concentration gradient of sucrose. So the water this unload will requires ATP. And after unloading sucrose, water potential of the sieve tube will increase back. And this water potential will drop. So the water will move up through osmosis from the sieve tube to the sink. And this lowers the hydrostatic pressure at the sink. And this difference in pressure will cause the fluid containing sucrose to continue flow from source to sink from here. You flow out back. Yeah, that's all for the recap. So for question 1 refers to figure 1.1, this figure. Name the structure ABC. So this, since there's no ribosome on it, so this is Golgi apparatus, and this is actually rough ER, and this one is mitochondria. State the name given to a region labeled D that separates the two sieve tube elements.